Hi, everyone, and welcome. We're going to let folks file in here. So we'll give everyone about a minute to join. I'm going to give everyone just 30 more seconds, and then we'll get started. All right, welcome everyone. We are right at time, just a minute after our scheduled start time. So I am going to jump in here. Welcome to everyone who's joining us and to our panelists. Today we're going to be talking about how data and technology platforms can improve diversity in clinical trials and clinical research. And I want to welcome our panelists. I'm so excited to be joined by such a wide range of backgrounds and current companies, roles, perspectives, and collaborators. I want to pass the microphone to each of our panelists to just provide a brief introduction about you, your company, and where you sit at this intersection of data, technology, and diversity, inclusion, representativeness in clinical research and clinical trials. So let's start with Pamela.
Hi, everyone. I'm so happy to be here, and thanks for inviting me here. I'm Pam Tenarts. I'm the Chief Science Officer at Meetingabol. We are a software as a service decentralized clinical trial platform to facilitate the use of decentralized elements to bring trials to patients faster. My background has been in clinical trials for 30 years, and I really am focused on bringing quality and efficiency to clinical trials. And decentralization is just one aspect that can help with that. Thank you.
Thanks, Pamela. And Nam, do you want to jump in with an introduction?
Sure. Hi, I'm Nam Nguyen, and I'm the Vice President of Medical Informatics and Data Science for the Life Science Group at Veridime. I'm also the Vice Chair of the Privacy and Security Work Group for the Electronic Health Record Association, and I am a certified high-trust de-identification professional. I've been transforming health data into insights to improve patient outcomes for over 15 years at companies like Medvantage, Practice Fusion, Allscripts, IMS Health, and now Veridime. So I'm excited to share what my team has been doing from diversity in terms of data today. And thanks for having me.
Great. Thanks so much for being with us. And Russell.
Hi, good afternoon, everybody. I'm Dr. Russell Robbins, the Chief Medical Information Officer at Purple Lab. Excited to be on the webinar and sharing some of the experiences I've had both here at Purple Lab as well as for the last 20 or so years doing health and benefits consulting as well as data analytics for a whole host of different systems throughout the whole healthcare arena. So thank you so much for having us, and we're looking forward to the webinar.
Thank you. And I should just end with, my name is Claire Crivero. I lead our public sector team here at DataVan, which means we see all of our team that services and works with our government, both state, local, federal government partners, as well as our clinical research and other research collaboratives. So eager to bring that perspective here today. And I think to begin with, I just want to give a quick overview of today's agenda and just a reminder about Q&A. As we move through this agenda, please feel free to drop questions into that Q&A function that you see in the Zoom webinar room. We invite you to ask questions throughout. We're going to answer questions at the end, and we'll get to as many as possible. And if we don't get to all of them, we will certainly follow up with you. And just to start with, so this overview of our time together today, we're going to start with a brief framing, and I'll lead us through that. And then we're going to go into deeper discussions with each of the panelists. And our panelists are representing two different sort of sets of tools in the toolbox for improving the representativeness of clinical trials and clinical research. So first, we'll have my colleagues from Purple Lab and Veridime discuss social determinants of health data and what that means, what that brings to this topic. And then my colleague from Meetingabol, Pamela, will chime in with the perspective on decentralized trials and how that is a tool that we can be using to increase the representativeness of clinical trials and clinical research. We also really want to dive into tangible outputs. I feel very strongly about this. I've been to a lot of webinars talking about diversity, inclusion, representativeness. It is a topic that is on a lot of minds right now. It's at the top of political agendas. We're seeing every agency in government. We're seeing a lot of companies in our community of practice prioritize this. I want to talk about the tangible outputs, not just the framing, but I want everyone to walk away from this webinar today with some really concrete examples of how we could put all of the we are learning here, these different tools to work to really move the needle on these issues. Just to jump in, just with the framing here, the reason that this is such an important topic, that diversity, equity, inclusion, representativeness in clinical trials and clinical research is so critical, is that ultimately science and innovation needs to benefit populations equitably. Currently, many products that are approved and are sitting on shelves in our houses and our local pharmacies, frankly lack the necessary evidence from a truly representative population, meaning that they have not adequately been tested in populations that truly resemble the populations who use or consume a given device or drug. This is a rising concern that bias is in fact baked into the scientific and regulatory process due to the lack of representativeness in this process of development of science. So I think that it's really important here that this is not just a talking about values or talking about doing the right thing. This is about the best science to inform the best products through the best regulatory process and the best scientific process. And the FDA has acknowledged as recently as last week that people from racial, ethnic minorities, and other diverse groups are underrepresented in clinical research. And I think this slide really hits home here, what that looks like, that we have trial participations that only represent 67% of the US population, that we have African American participation like very low as compared to the population in the United States, that in clinical trials, Hispanic population is at 1% versus the representation in the United States in the general population. These are critically low, and we really need to be thinking about what all of our responsibility is to move the needle on improving this representation. And I think that it's important to, on that right side of the slide, to be thinking through what these levers are. What are the issues here? What are the obstacles that we are facing as an industry when we're thinking about increasing the representativeness of our clinical research, of clinical trials as well? And I think we talk about the source data a lot, garbage in, garbage out, that social risk factors may not be recorded in the structured data that we're currently using or in the traditional data sources that we're currently using. De-identification, and we care a lot about that at DataVan. De-identification may be applied to real world data in a way that erases variables that are critical to measuring diversity and inclusion in our clinical research and trials. So we've gathered here to talk today about the expanding toolkit. And we should note at the top that there is no fix, there is no one panacea, there's no hack, there is no technical innovation that's going to solve everything on this slide. But everyone who's spending time preparing, attending, and delivering this webinar is committed, I believe, to figuring out the levers we have to do better as an industry. Not just because it's the right thing to do, as I said, but it's the right thing to do for better scientific process, for better products, safer devices, safer products that we're going to find eventually on our shelves. So one major piece of vocabulary that I want to address up front is SDOH. These are social determinants of health. And these are the conditions in the places where people live, learn, work, and play that affect a wide range of health and quality of life risks and outcomes. There are 5 major areas. These are all defined by the CDC: healthcare access and quality, education access and quality, social and community context, economic stability, and neighborhood and built environment. When we talk about diversity, inclusion, and research, we're not just talking about race and ethnicity, we're talking about the intersecting and multifaceted aspects of all of these social determinants of health. I'm an epidemiologist by training. This gets really hard and really complicated, and we're talking about the layers of vulnerability or the layers of identity that someone may have and may be bringing to the scientific process. There is growing awareness that these factors, these social determinants of health, need to be considered when we think about representativeness in clinical research and clinical trials. And in my role at DataVan, sort of facing the public sector, we're seeing momentum and growing interest across sectors in connecting this valuable data to some of those traditional data resources. So just a brief moment, if those of you are not familiar with DataVant, we work to reduce the friction of data sharing across the healthcare industry. We're working to build this neutral, trusted, ubiquitous data connectivity that protects the privacy of patients while supporting the exchange of identified and de-identified health data across tens of thousands of healthcare institutions. In the public sector, we have seen in our piece of the connectivity puzzle, we have seen a growing interest from our government, our academic, and our commercial partners to connect the traditional sources of data to SDOH data. For retrospective observational research, we've seen this in the COVID-19 database that we support. We've seen this in our clinical research networks that have connected different academic medical centers for bigger, more centralized data, and they're supplementing that connected clinical data with SDOH data. And we've seen government institutions like the NIH and other agencies think about how they can be enriching traditional data sources with SDOH data. So then turning an eye towards prospective research and clinical trials. Just last week, the FDA sent a very much anticipated draft guidance document on the topic of enrollment of diverse populations in clinical trials. It's now sitting on the desk of the White House's Office of Information and Regulatory Affairs for review. And this is really signaling what, the guidance and policy that's going to shape our industry. And frankly, we, as I said at the top, we should not just be responding to these regulations, we should be responding to a representative scientific process. So I think we're going to see more momentum on this topic, but I think the fact that you are all here today means that we are not just waiting for that guidance, we're not just waiting for policy to catch up, we should be leaning in and figuring out what the tools are to improve those factors that we went over at the top on that slide before the regulations or before that guidance document hits the FDA. So for a moment, I want to hear from all of you, our audience. So we're going to do a quick poll. We've gathered here today about learning about the tools that can help us improve diversity in the representativeness of scientific research and discovery. So I'd love to hear from all of you. What data and technology platforms are you using to help diversify trials and research today? We'd just love to hear where we're starting, where our baseline is with this audience. We'll give folks a moment to answer, and please choose any of the following, and it's a multiple choice question: innovative patient data sources, advanced data analytics technologies, decentralized trial platforms, other data and technology platforms, or no data and technology platforms implemented yet. You're here at the baseline, here to learn. All right, I think we're going to wrap up pretty soon. Just get those answers in. Okay, interesting. We have a really wide range here. So we have some folks who are coming here that we're implementing innovative patient data sources. It looks like we have quite a few people with no data or technology platforms to date. So really, really thrilled that you're with us to learn more. Fantastic. This is very helpful for our panelists. So now we have a little bit of a sense of our audience as we move into the next section. So we're going to start with social determinants of health. With my colleagues, we're going to talk about this novel data source and how it can help us. So I'm going to pass the mic to Russell to kick us off.
Thank you so much, Claire. Hold on, let me just pull up my slide here for 1 second. Okay, great. So at Purple Lab, what we have done over the last several years as we're building our Health Nexus platform is we're really looking for all of the different sources of claims information that we can have to build our models, to put into our data warehouse, and to use for all of our analytic purposes. So we get medical and pharmacy claims at the transaction level. We also have over 25 million individuals who have died during the course of the data construction. So we can do longitudinal studies in that regard. We've also taken into account other types of government programs, whether it's CMS's hierarchical condition categories or HCCs, risk adjustment factors, other types of programs, looking at the differences between the under 65 and the over 65 populations with regards to risk and to other types of condition categories, and looking at all of those individuals and putting them together and stratifying, and then overlaying on top of that the social determinant of health information. So that way we can really gain much deeper insights into an individual with regards to socioeconomic, race, ethnicity, and other types of factors that might cause them to have some type of other encounter within the healthcare system. And we'll be showing some of our findings from that later. The other thing that we've also done in our data set is we have taken all of the providers that we have, we've mapped them with their NPIs or national practitioner information, so that way we can identify each individual provider. So while the patients are all de-identified in our data set, through using of tokenization with our partnership with Datavan, through other types of information sources that we get, we can actually identify or the de-identified member with some of the trends, so we can then report out at the 3-digit zip code level what's happening both for the providers taking care of those patients as well as other trends. And we can go to the next slide. So as I mentioned, we've built our Health Nexus platform using de-identified medical data, using pharmacy data. We've put that together into 2 different ways so that the user can either come and directly query into the data itself to answer specific questions, or to work with us to generate customized reports. So that way, if you want to have specific questions answered and you don't have the staff to do it, we'd be happy to do that with you to make sure that it all works. And so what are these insights used for? It's really looking at patients, providers, suppliers of all sorts of different levels, and really just looking at the data in a very new way than it's been traditionally done. And that's really through using the social determinant information, because with that overlay, it's no longer just a population of diabetics, but we can now stratify them into different groups, into different ways that their treatment is being rendered. So we can now understand at a much deeper level what's happening to sub-pockets of that population. So it's no longer just the disease itself, but what's happening to individuals who are in that phase of the disease process. And I think we can go to the next slide. And so, what we've done also is, as Claire had mentioned at the beginning, there are those 5 categories from the CDC on social determinants, but through other partners and other relationships, we've really made it into a much more detailed type of matrix. And so with that, we can really look at all sorts of different factors: income, age, gender, and the combinations of all of those different things together. And later on, I'll be showing some a study we did with a pharmaceutical company, looking at how their drug was being not only dispensed, but what were the denial rates, and how did those differences occur in people with different marital status, different socioeconomic status, different race and ethnicity. And so what we're finding is, it's no longer just that the drug was filled or not, but why wasn't it filled? And who do we need to start reaching out to more to deal with those inequities and variables in the system?
Thanks so much, Russell. And I'm interested just for our audience, if our audience are coming here and thinking about trial design and their recruitment processes, at what point in the trial design, at what point in recruitment processes do you recommend a trialist, be them life sciences or other investigators, plug into the Purple Lab data and platform? When is this data and platform most impactful in order to expand the representativeness of their work and the science powering their pipeline?
No, that's a great question. And what we find is that, as early as possible in the process, because what happens a lot of times is somebody has a question that they want to investigate, but as they start working with us further, they discover that their initial question may need to be tweaked a little bit. There may be other fields or other parameters that we have that they hadn't thought about. So it may expand what they want to do initially. And it's really just that collaboration that we find to be so useful to make sure that the specific question is being addressed and answered. So as early in the process as possible, but really at any point in the process, we'd be more than happy to help out and make those things work.
Thank you so much. And I'd love now to transition to Nam to join the stage and talk a little bit about the Veridime perspective on SDOH data.
Sure. Before diving into how Veridime can be leveraged to improve diversity, I'll provide a little background on the company. So some healthcare technology companies can deliver real-world clinical data. Some can help turn real-world data into insights, while others serve providers directly with point-of-care software. What Veridime does, it sort of does it all, making the most of our unique position at the center of healthcare. We drive value through our scalable network, called the Veridime Network, which brings advanced capability and data-driven solutions to help transform healthcare. One way the Veridime Network addresses diversity for life sciences is by allowing us to link Veridime data with our partners' data sources in order to improve both data content and geographic representation. If we do not have the data points or patient representation a study needs, then we can look to our network partners for data to link to and fill in those gaps. Integral to the Veridime network is our provider network. Veridime is a wholly owned subsidiary of Allscripts, an EHR organization, giving us direct access to sites that utilize the Allscripts family of EHRs, such as TouchWorks, Professional, and Practice Fusion. This also means that we have access to the connected data of the labs, prescriptions, and other clinical workflows from our site's EHR experiences. With this bidirectional data exchange through our network, we can provide real-world data for both retrospective and prospective studies directly from the point of care. Couple that with our research consulting services, and you have an end-to-end solution where we can bring timely data from the point of care directly to research. Next slide. Veridime's current network is nationally representative and contains over 180 million patients. 45 million of those patients are linked to closed claims through the Veridime network, giving you more complete picture of the patient journey. Because we have a direct access to the sites through our EHRs, we also have access to unstructured data, such as physician notes, attachments, and images. The real-world data studies using this data have better representation of populations by geography and a more diverse population of patients to base studies from. It also means direct access to data from the point of care that other real-world data providers generally do not have access to. However, we don't just address diversity by providing real-world data. Once again, with our direct access to EHRs, we can target and recruit sites and patients for prospective studies at scale within the Veridime network of providers. This is Veridime's study source solution, which provides researchers access to novel sites not traditionally recruited from. Recruitment today is done heavily through large academic medical centers and large hospitals. However, adding Veridime to your recruitment strategy gives you access to our network of solo practitioners and other small, medium, and large practices from rural, suburban, and urban settings. This increases your access to more diverse population of patients. We're currently working with select partners on proof of concepts, which are recruiting sites through the Veridime network. Next slide. For our real-world data users, Veridime curates this data and makes it what we call research ready. For those that have used EHR data before, it can be messy, inconsistent, and difficult to use. For those that get real-world data directly from Veridime, a team of clinicians and informaticists have curated and harmonized the data, making it fit for purpose for research. Also, since we have direct access to our EHRs, the data is longitudinal and it's very timely. For some of our data, for example, the data is just weeks old instead of months or years, because it's coming directly from the point of care. Our EHR connection also gives us the ability to prospectively capture data, including social risk factors, directly from providers at the point of care through our EHR and other technologies. So our prospective data capture can potentially gather social risk data gaps directly from sites. Now, let's talk NLP. For select sites, we can use natural language processing to mine data out of free text to integrate and enhance our research-ready data sets. A lot of useful information is in narrative notes that providers simply do not structure, and you can only get this information from a data partner that has access to the source. We've done NLP for a variety of therapeutic areas, as you can see here. Bone mineral density values went from barely 100 data points to over 80,000 values using NLP. HbA1c values increased by 32%, adding over 2.5 million data points to use in research. And in terms of social risk factors, we were able to improve data points on intimate partner abuse from 13.5 thousand data points to over 300,000 using NLP. I'll go over more social risk data that we've mined later in this talk. Finally, my last note is about de-identification. For those in the clinical research space that need to work with de-identified data, it's important to note that social risk data may be erased through the de-identification process. As an example, many racial minorities, such as Native American, are erased and generalized to other in the de-identification process. So even if you have permission to use identified patient data in your study, if you link to a partner that only can provide de-identified data, you may not get the granularity in social risk data like race that you were hoping for. But with Veridime's access to source data, access to the nation's top de-identification experts, and control over our de-identification processes, we can potentially customize data sets to compliantly fit your research goal, especially if you're including sensitive data like social risk factors in your studies. Back to you, Claire.
Thanks so much, Naman. As a researcher, perhaps a former researcher, I don't write many papers these days, but hearing a little bit about these variables that you're finding from notes that you are all able to retain as part of the data sets given your pipeline data is super interesting and very important as we think about linkages, particularly for clinical research, clinical trial design. So before we move on to the decentralized trials and hearing more about decentralized trials, I just want to pause for a minute and trigger just another poll to learn a little bit more about our audience and their the issues you have all faced, any challenges you have all faced with SDOH data. So for this poll, I'm interested in what challenges are you all facing related to using innovative social determinants of health data sources for insights into diverse populations. And this will hopefully help us as we come back to concrete examples. So interested if any of you are assessing which data set is fit for purpose, if that's a particular challenge, finding the right data partner. There's a lot of SDOH data out there. Is that a challenge? Ensuring patient privacy and HIPAA compliance. I think as Nam touched on, making sure that you're rigorously using data, but doing so in a completely compliant way. Matching patients across data sets to find cohorts of interest as we supplement this data, how can we find these sub cohorts? And getting organizational buy-in. This is a new type of data. As an EPI, I mean, we're really used to the Framingham data source, the nurses study. Using this new SDOH data is new for a lot of us. So interested in whether we've faced challenges with organizational buy-in. Okay, so super interesting here. Okay. It looks like a lot of folks are just trying to figure out which data set is fit for purpose, and fit for purpose is a big question. I mean, and sometimes I feel like fit for purpose is a little bit in the eye of the beholder. There's different frameworks for determining fit for purpose, and maybe we can return to that later. Figuring out the right data partner, and I hope today we're helping a little bit with that, of figuring out the different partners at the table. Ensuring privacy, it's good to see that's not in the top. I'm glad that everyone's thinking about privacy and thinking about compliant ways to link data. And fantastic. And matching patient cohorts across data sets. Super interesting, getting buy-in. Very, very interesting. Thanks, everybody. So we're going to pause on SDOH. We will come back to concrete examples. But for the next slide, let me make sure I can advance. I'm going to pass the microphone to Pamela. In addition to layering on important data, as we talked about SDOH, there's these operational improvements that we can implement to increase the representativeness of our research, of our trials. So Pamela, I'd love to invite you to the stage to take the microphone.
Well, thank you so much. And yes, so with Meetingabol, We strongly believe that research is for everybody and every biology. And what we mean by that is that health and representation in trials is a human right, and participation should be easy. The history of randomized clinical trials is that it gives really good evidence, except it's only for the people that were in the trial, and it's very hard to extrapolate that. And so to have good evidence for everybody, we need to have representation in those trials of everybody. We've not always done that very well, but inclusive medicine and decentralized trials are maybe removing some barriers to that clinical trial access, while also maybe offering clinical trial efficiencies. And those efficiencies accelerate science, faster cures, and better represented cures, and they support collaboration. So Meetingabol is a decentralized clinical trials platform. I saw in the chat earlier to sort of comment on whether it's US only or global. So we're global. We've had more than 1 million people, I'll say, interact with our platform, over 60 countries, over 60 languages, and over 100 clinical trials. So we've done a lot of these things, and we've done them pretty much everywhere. Before we go forward, I want to give a little bit of an explanation as to what we mean with decentralized clinical trials. If we can go to the next slide, please. And I'm going to use the definition of the Clinical Trials Transformation Initiative. I used to run that organization for 10 years. It's a public-private partnership between the Duke University and the Food and Drug Administration. It had a very specific mission of improving quality and efficiency of clinical trials. And one of the ways we thought that could work is by using more decentralized elements. And the definition, this is 2018, so they're updating it right now. But at the time, it was defined as trials that were executed using telehealth and telemedicine, also including mobile and local healthcare providers. By the way, some of those healthcare providers, the local ones, we used to use those all the time in clinical trials. We used to use local labs and things like that. Then we went to central labs. So this is going back to local providers, and then procedures that are a little different from the traditional brick and mortar trial model. Maybe the investigational medical product is directly shipped to the participant. Maybe we ask for surveys and clinical outcome assessments to be completed over a tablet, a telephone, things like that, to sort of really bring the research to where the patient is, rather than having the patient have to go to the research site, because that was where the trial was. So we're trying to bring clinical trials closer to the participants and closer to the sites. And the benefits of using these modalities was really that we thought it could facilitate faster clinical trial recruitment. It would be in better retention. It would be easier to stay involved in the trial. It would be a greater control, convenience, and comfort for the participants. We all know that 70% of US people live more than 2 hours away of a big clinical research site where they could come for research. And a lot of people just can't do that. It doesn't fit in their days. They can't do it. And this, hopefully, by using these modalities, we would improve participant diversity. If we go to the next slide, I want to talk a little bit more about what that looks like. So thank you. It's really needs to be fit for purpose. And when I say the word decentralized clinical trials, it's everything from a direct-to-patient clinical trial to using just a few of these modules or whatever version of the combination in between. So again, it could be remote screening, e-consent, consent done over a 21 CFR Part 11 compliance system, the ECOAs and EPROs that you can complete on the phone, like you see here. You also have a screen here of what a televisit would look like. You would have connected sensors, so use weight scales, use heart rate monitors and blood pressure monitors to sort of have that data directly flow to the databases of the sponsors. And again, have the ability to include local physicians, home health, and labs. And the Meetingabol platform combines all these data flows into one platform that makes it really easy for everyone to participate. If we go to the next slide, what I want to show here is a slide that was shown by Sharday Arrojo, who's part of the Office of Minority Health at the FDA, showing the guidances that are already available. Claire mentioned another one that's coming, but there's already a lot of work being done to make sure that diversity of clinical trial population represents the people that ultimately will benefit from the medical products that are coming out on the backside. With Citi, we also had created a Transforming Clinical Trials 2030 vision. And what we said there is that clinical trials should be patient-centered and easily accessible, and the enrolled participants have to reflect the diversity of the population expected to use the medical product. So this is all coming together. MRCT has done a lot of good work about what the specific data categories should be, because that's another thing in clinical trials. We all call things different names and we collect it differently. And they put forth a framework for all using the same verbiage, like using the same race categories, the same ethnicity categories. So you can put this together in a harmonious situation, whereas now people have to retrofit things together. And if we go to the next slide, I want to also point out that we've talked a lot about technology, and that is great, but usually technology doesn't do it on its own. We really need to include people, and Nam talked about that a little bit, the community providers in clinical research. We need to not only conduct trials at the big NCAT centers that are sort of around the big population centers in the United States and a little emptier in the middle. There's a lot of people that live in the middle of the US. They need to be represented. It also is rural populations, urban populations. We need to reflect all the populations that are there, and to sort of not only think about data and technology, but make sure that this is that we involve the community providers. Again, that we make it easy for the community providers. That could be where using available data sources, using technology makes it a little bit easier for my primary care physician to participate and not be overwhelmed by all the research tasks. So I think as a last slide, I would like to just point out that we just had some research news that came out. We worked with Tufts Center for Drug Discovery. And what they found is that if you invest in these technologies for decentralized trials, you actually get a return on your expected net present value. So if you do it in phase 2, it comes back at you 5 times. And if you do it in phase 3, the value of that investment comes back in multiples of 14. So it might cost a little more upfront, but the benefits you get back are really multifold of that. To sort of also say that it's worth doing these investments, because it is a little different and a little more than what you would normally think of spending. But hopefully, over time, even those investments will come down. And with that, I'm going to turn it back to you, Claire.
Oh, thank you. And I think 2 things. First of all, that point on there's a lot of people in the middle of the United States. My husband from rural Midwest, 2 rooms over, can hear him silently punching his fist in the air. I think that's a really good point, that of all of our trials, if we have a center of gravity in Boston, what are we missing when we miss these populations that are not adequately represented at these huge trial sites? Super interesting. I think this last slide, for those of you who signaled that you're working on organizational buy-in, this slide, I think that Pamela shared with us, is like, make sure you take these numbers with you into these conversations, but this is a good decision for science. It's a good decision for an investment, an upfront investment, thinking through how we can be investing in these tools upfront and what that actually means in terms of return on investment. I think this slide really captures that. If we need to make a case to our organizations about why it's not just about science and validity, it's about a good business decision for your product development and your pipelines and your research. So I think, as I promised, I really want to dive into the concrete. So over the next 10 minutes, I'd love our panelists to highlight some of those real world examples. And this actually has come up in the questions already, thinking very concretely about how these tools can bring impact on the subject at hand of increasing that representativeness of our clinical research, of our trials. So I'm going to start with my colleague, Nam.
Thanks, Claire. Veridime has been working with academic partners on understanding what social risk factors physicians are capturing in our EHRs today and how our sites capture them. This is being done in 3 studies. The first study created a framework for extracting and reporting of social risk factors from ambulatory EHR data. The paper was published at AMIA last year and provides an open source Python dictionary to facilitate the reporting and extraction of social risk factors that anyone can download and use. Our second study, which we're currently finishing up, use that framework, and we'll describe the social risk factors we found in structured and semi-structured data using 10 years of EHR data spanning 3 separate EHRs. And our third study, which we'll work on later this year, is to dive into physician notes and chief complaints and other unstructured data and see what additional social risk data we can find and complete our understanding of the current state of social risk factor documentation. Next slide. Here, we're showing stats from our second study. This is a distribution of our study data by race that have social risk factor data mined from EHRs. What jumps out to us here is actually the amount of non-demographic social risk data that is currently documented today, but really only available to those that have direct access to the source and have the skill set to mine them out. Next slide. And here's more results from our second study. You're seeing the top social risk factors mined by prevalence. We see that practitioners are organically documenting information on housing issues, unemployment, intimate partner abuse and violence, issues associated with social isolation, as well as stress. So again, this is the type of social risk data only available with direct access to data at the point of care. Back to you, Claire.
Thank you, Dominic. And what strikes me on this slide, I think this right at the top, that disappearance or death of family member and how that might be contributing to social risk. I mean, this is on the front page of the news. This is in the literature. It's in the pre-publications and the peer review publications that we're seeing that particular one more and more when we think about stress, when we think about populations and making sure we're adequately accounting for social risk. I think that one in particular jumps out to me as a really important one. So thank you for sharing that and how you all are getting that richness from the data. So I'd love to transition to Russell, to Meetingabol, I believe, to Pamela to talk a little bit about this.
Yeah, so the 2 studies I'm going to be talking about really highlight how you can get closer to potential participants. So in this particular trial, it was a rare disease, a biotech company that came to us. And obviously, as the name implies, rare diseases are rare, and the participants are probably not conveniently situated next to a clinical research site. So what we did here is we recruited through email, and then the app was downloaded, and we could do that all online. Once a patient was recruited, they would complete an e-consent online and an enrollment questionnaire. And once they made it into the study, they would get a welcome kit. And with that kit, they could visit local labs. The labs were reviewed for eligibility, and then the participant met virtually with the sites and completed ePros. This example is a complete direct-to-patient example, and I think both of them are. But there was a question earlier about whether DCTs were applicable in all clinical trials, and I would say they are. Every clinical trial can be made a little bit more patient-centric by making it easier for the patient to participate in them. But obviously, some trials will require still visits to an on-site center, whether that be a hospital or research center, if there are anything that needs to be done that cannot really be done remotely, obviously. So, but we believe that every trial has the opportunity to have some elements, and that's why I was talking about the fit for purpose. If we go to the next slide, it's another example of how you can get closer to the patient. This was a psoriasis study, a registry. And what we did here is we, obviously due to COVID, and a lot of decentralized elements have become a lot more adopted during COVID. I probably would have to say that. But during COVID, they needed to move to a more flexible and a remote deployment to be able to continue with engagement of the participants and the data collection. A lot of people were told to stay at home. A lot of people did not want to come into research sites because that's where it was perceived the virus was. A lot of sites closed because they were not deemed to be mission critical when the hospitals were full of COVID patients. So this protocol fit solution worked within the existing protocol. So not maybe the most ideal situation. You probably want to think of these elements ahead of time, but we allowed for the physician to enroll a participant into the trial by engaging them in the app. Then they would complete again an e-consent and an e-pro. It would be reviewed by the site staff, and there would be continuous outcomes data collected, and then the payment would be triggered. And what we found here is we could easily change over traditional processes to remote. People told us that they liked it, and it accelerated things. But what I think is most critical is that we were able to reach more people, and more people means also more diverse people. So with that, Claire, I'll turn it back over to you.
Oh, thank you, Pamela. I think it's important to flag that I think COVID-19 really accelerated that there was a real need to make sure that people didn't need to travel long distances, go in person, and that there's been a real acceleration of decentralized trials. And as we think about pandemic preparedness from a public health perspective and future-proofing trials, thinking about this as a tool in the toolbox of not only expanding diversity and inclusion, making sure people can access clinical development, clinical studies, but also thinking about future-proofing and pandemic preparedness of this being a tool in the kit for resiliency. Super, super helpful. And Russell, I'd love to pass the mic to you.
Great, thank you so much, Claire. So, one of the things that, as I've been thinking about this is, we look at the outcomes and we look at who's using drugs, who's not using the drugs, but also thinking about it in terms of, clinical trials is making sure that you have this type of information at hand. So as you're recruiting people for the trial, you're making sure you have that wide representation. And so what this study here is, it's highlighting really some differences that we were noticing with a particular pharmaceutical agent. And we were looking at 3 broad categories. How often was this drug dispensed? How often was it, what we call reversed, which really is abandoned. So the patient goes to the pharmacy, they fill out the script, but then they never pick it up, for whatever reasons. How often does that happen? And then how often does that happen, where the patient is just having the drug outright rejected because of plan design or other types of factors that, make them unable to obtain that, fill that prescription. So when you start looking here, this is really just a simple graph of age. As people get older, the number of people who are able to get this drug in, changes over time. But what we see is that if you're earning less than $20,000 a year, you are going to have more rejections, but fewer reversals than those who, you're going to have, if you're making more than $20,000 a year, you're going to have more rejections, but fewer reversals than those making under $20,000. And if we go to the next slides, as we start breaking this down further, what we can see here is this is just an example of ethnicity looking at Hispanics versus non-Hispanics. If you are within that dashed line in the middle there, that means there's no statistical significant difference between you versus the other category, which in this case would be the non-Hispanic population. However, what we see here that the odds of Hispanics are 1.25 times higher among all of those who, for this same drug, are in that rejected group. As we move to the next slide, what we start seeing now is what is the impact of race? Well, for African Americans, it's 1.5 times higher than the non-African American population. So again, the reason for having the drug rejected is greater for certain for African Americans. And then if we go to the next slide, what we start here now is combining race and income, and all of a sudden it goes from 1.25 to 1.7 times higher for African Americans who are of low income having the drug rejected than just African Americans or the control group of everybody else. So there really is some differences as to why the drugs aren't being filled for this group. And that's one of the things that we have to explore further. Is it something in terms of prior authorization? Is it something at the pharmacy? Is it a benefit design question? I mean, these are just, questions that we don't have the answers to from the, directly from the claims, but ones that, we want to alert the, in this case, the manufacturer of this pharmaceutical agent as here are some problems with, the drug being distributed, and maybe these are types of things that they want to do further outreach to the provider, to the patient community, to other communities. And we're seeing this from other organizations as well, who might have access to, other subclasses, LGBTQ, or other types of groups that aren't in the traditional data capture. What are they seeing, and how are they also finding this data so we can work with them to understand some of the disparities and inequities? And if we go to the next slide, what we can see here now is if we start combining everything together, for the African American population that is low income but is married, while there still is some of that higher rejection rate compared to the other to the rest of the population, what we're seeing is a little bit of that is mitigated. And again, what the impact of marriage has on that in terms of, making sure that you're getting your prescriptions, if you're leaving them at the pharmacy, is it your spouse reminding you or your spouse going to pick them up? There may be other reasons why being married can change some of those factors around. And also just is being married in itself, mean that you may have other resources to, to lower some of that rejection rate. That's one of the questions that we still want to try to answer and address. And so that, Claire, I think takes me to the end of, this little case vignette for this drug. But, we can find these types of things for really any of the drugs that we look at, for any of the different types of care that we're looking at is, these social determinants really do play such an integral role. And for so long, they've just been either ignored or not really addressed. And now that we're finally coming to the point where we need to start looking at it and addressing it, is how do we make sure that we're doing it in the most, efficient and effective manner as possible?
Thank you so much, Russell. And I think it's a very important point. And I think stratification, as an epidemiologist, this is so critical. What you can only stratify by what you have in a data set. And there's all kinds of unknown confounding, unknown things you can't control for when we don't have this kind of these variables incorporated into the data set. So I so appreciate you highlighting that. And I think it links back to what Nam was saying too about how Veridime has thought about this and how you are all thinking about this. I think it's really critical as we think about our research design. How are we incorporating these important factors? Because to your point, they have been overlooked. To your point, they are not incorporated in models. They're not incorporating all the AI and the machine learning we want to build unless we are able to capture this granularity. There will be bias baked in. And as we move on from these really important vignettes, these concrete examples, we're going to move into Q&A. And I've been monitoring the Q&A from everyone. And please feel free to throw more questions in there. And let's start with SDOH, and then I have a few questions for the decentralized trials. There was a great question about LGBTQ plus individuals, and thank you for jumping in and answering that one. And I think there's been some interesting questions about SDOH data types and variables and what's available in the variables. If you could chime in, maybe we'll start with Nam and also Russell. There was questions about how race is coded. There's obviously, and Pamela touched on this, there's so many different frameworks for categorization. And often what we've seen in traditional data sources is this giant lumping. And there were a couple questions about how are we disaggregating these groups? Are we using frameworks? How are we thinking about these really complex categorization? And is there anything that the audience should know about that race, ethnicity, variables available in these data sets and how they might inform research? So now I'm gonna start with you and then we'll pass the mic to Russell.
Oh, sure. I'm happy to sort of answer the LGBTQ question. So as part of electronic health records, I think 2015 meaningful use happened, and in that the HHS mandated that each EHR that wants to be certified has to be able to capture sexual orientation and gender identity. Of course, a lot of those didn't get implemented until a few years later, but by now, all modern EHRs should have the ability to capture those fields. The issue in terms of use for research is that this is a very sensitive data field, right? It's just one of these hot topic, hot button patient identifiable, potentially triangulation type things. So this is one of those cases where the data is being collected. We don't know exactly how well the data is being collected, but it's very unlikely that you're going to find this data in just readily available data that's out there to link to today. This is, goes back to the case of, you really need to find a data partner that has access to the source and has, the expertise in de-identification and compliance of that data to make sure that you can use that data compliantly. But it's definitely out there.
Wonderful.
And the thing I'd like to add to what Nam just said is also, one of the challenges in terms of claims adjudication at the health plans is, a lot of the edits that are in place to say that this claim is, not applicable, is, shouldn't be paid. A lot of that is also changing. And so the plans are also trying to readjust. So if you are, self-identified male, there is still a possibility you could become pregnant. You could, so those claims would still have to be paid for, except, the claim systems don't traditionally allow when you just had a binary type of thing of male or female. That was it. Well, now that the plans themselves and the industry is recognizing that, there is much more that needs to be captured. It's how do you deal with that type of information as you're processing and paying, and then on the back end, analyzing that type of information. So, so yes, it is a very sensitive topic, but it is definitely one in the forefront that, people are addressing now.
And I would also talk about one of our audience members made a really astute comment in the Q&A, which was there's a difference between ability to capture a certain variable, then it being captured, and then captured well and regularly. And I think that's something that we're also getting to when we think about these hot topics, these new variables that we're recognizing are really important to incorporate, but perhaps weren't in the original structures or weren't captured well in previous structures, that this distinction, ability to capture, being captured, and then captured well and regularly, I think particularly is important. Thinking about legacy systems, thinking about data source selection, really astute observation. I think that's an important one, and keeping an eye out for those distinctions is important. I'd love to just talk a little bit about decentralized trials, a little bit, Pamela, turning to you for a moment. And I think there's a very straightforward question, and I'd love to get your perspective on it. Does decentralized trials design, has there been evidence that this is getting viewed very favorably in grant applications and implementation? Do we have any evidence that grant applications and other very tangible measures of success of being able to move a trial forward, that DCT is really helping move the needle there for folks who are designing trials?
So I'm not sure I'm going to have a definite yes or no on that. No surprise, I imagine. And I'm sure it depends, it depends on the execution, on the trial, on the platform. What I will say is that in 2018, I think it might have been 2019. I get a little confused as to what the time frame is of anything before. It's like before COVID and after COVID. Before COVID, the NHLBI had a big workshop where they brought in different digitalization elements of clinical research. And they brought in decentralized clinical trial experts, people that had done, Apple Watch studies, people that had done other studies. At the time, I was still at Citi. And they ended up writing that all up in a publication that I think is called Digitizing Clinical Trials or Digitizing Trials. And many people were part of it. Craig Lipset, who is one of the evangelists around DCTs, Rob Califf, Harlan Krumholz, like literally a lot of people. And what I'll say is that to me shows an appreciation of this going forward, and shows a willingness for it to be for decentralized elements and digital data approaches, combining real world element data into clinical trials, for that to be considered. Again, it has to be well executed. It has to fit the protocol you want to do. So while I cannot say that, yes, you're going to get brownie points for putting in decentralized elements into your grant, I think if you execute it well and it makes sense in your application, that there is an appetite for that. Is that going to be across the NIH? That I don't know, but I would imagine. And I know some, like it has to be at NIAID, because some of the vaccine trials used an ECOA on a bring your own device. So that is, and that was approved by the FDA. So I have to think that there's a lot of more appreciation for this right now. It has to be well designed. You have to show that you haven't degraded the data, obviously, because involving technology, now you may have systemic issues in data collection, which is a great thing if you can make it easier, maybe not so great if a mistake creeps in there somewhere. But I would say that there's, I mean, everybody knows, and we all watched the presentations at that workshop about, you know, everything is online right now. Why do clinical trials still have to sort of pretend as if the digital world doesn't exist? So I think well executed, fit for purpose, doing it right, a good argument for doing, I think that is going to be well received.
Wonderful. There are many more questions than we have time for. I want to help get us out right on time, as I know everyone likely has back-to-back Zooms in this audience. I want to thank everybody for joining us today. And I'd like to leave us all with questions for our audience as well. Taking the toolbox here, taking these concrete examples, I think I'd like to challenge all of us to think about the concrete things we can all be doing as a community of practice to move the needle on representativeness of our work, representativeness of our trials, of our scientific development, of our research. I think you have a few examples here. We'll make sure, and Chris has said, like, making sure slides, and this is available to your organizations as a recording. We hope that you will share the news on this toolbox, that we can continue this conversation, and we can all be thinking very critically about how we all move the needle forward using some of these tools, using some of these concepts that were introduced by our panelists today. So to Nam, to Russell, to Pamela, thank you so much for joining us. Thank you for this thought-provoking presentation, and eager to follow up with everyone who's asked questions. We'll make sure that we reach out. Thank you so much. Have a lovely day, everyone.
Thank you.
Thank you.